advocacy groups that are online and supporting us and our other research partners. And what I'd like to talk to you about today, this evening, is how we can start to translate the MRI science into accurate enough sort of details such that clinicians and researchers have a way to sort of utilize the MRI findings. And I really want to stress this all throughout that, what we're discovering and learning about are patterns of kind of injury and disease progression. Um, and it's, this focus is really trying to ready ourselves for even better studies and hopefully laying down the foundation for developing treatments. Um, and in my case, not that Elizabeth left this out, but I've had the amazing opportunity here at Georgia State, here in Atlanta, where I was able to work with 58 of our patients and our families in essentially assessing our baseline visits. And so, quite honestly, that's been my favorite part of being involved in this project. And so I really thank you all for that. It was great to be able to experience and participate in that. Um, so so why are we here? You know, out of the gate here, I just want to acknowledge that we have patients and families here. We have neurologists and trainees and other clinicians, and we have advocacy groups and, potentially even trial stakeholders. And so what I want to try to do is just keep the language that I'm using accessible to everyone, but also keep the science accurate and more, and most importantly, just keep the why like visible to all of us. And so I want to just to kind of outline just what I'm going to talk about here. I want to talk about just the CADASIL big picture and then specifically anchor the white matter hyperintensities or lesions as our sort of anchor marker, but also talk about other MRI markers that we're actually collecting and just how that all fits together and what that looks like in terms of symptoms, progression, and again, relevance for any future clinical trials. So CADASIL imaging is not just about the pictures that we can generate or the features or the things that we can measure, but it's really about understanding the people and your families sort of over time. So CADASIL affects really all of you, and it's over many, many years. And MRI can reveal and expose potentially some of the aspects of the disease burden, but and some of that is apparent before any symptoms are obvious. And so as we continue to improve our imaging markers, we want to essentially, as best as we possibly can do, we want to characterize these over time so that they can be used as clinical trial endpoints. And so I think I want to start out with, but not get too technical and too into the weeds, if you will, on just like the pathophysiology of CADASIL itself. But I do want to just show this slide and talk about this a little bit. And this is not my slide. I actually have the paper I've cited here just because they did a great job, and I could try to draw this up myself, but I'm gonna just not reinvent the wheel here. But as most of us know, CADASIL is a genetically autosomal dominant, genetically determined small vessel disease, and it affects the brain's small vessels in a very distinct way. Um, these vessel changes then contribute to the tissue injury, and then eventually the symptoms that are sort of observed over time. And so uh, the Notch3 mutations affect uh, the vessel wall proteins, and mostly this is affecting the permeability of the vessel wall itself. And over time, um, this these sorry the protein mutations actually cause the degradation of the um, endothelial cells, the astrocytes, and the you know increase the permeability, if you will, so the space in between. And then also the death of these cells essentially leaves part of the vessel wall just exposed wide open. And so
Jeremy, I'm going to interrupt you just a
yeah.
moment. I'm not sure if I need to advance your slides or if you
Oh, I'm
want to just
sorry. Yeah.
say next
Oh, that's
slide when you're
helpful.
ready.
I'm on slide 3,
Okay, thank you.
and I'll
And
just
then
say
just say next
next
slide and
slide.
I'll keep it moving.
Yeah, yeah, gotcha. Thank you for that. So as the support cells around the vessel wall degenerate, the vessels themselves become less able to regulate blood flow. And thus, because the blood flow is what supports and protects the brain tissue, this is what leads to visible white matter injury. And then we'll talk about what that looks like in terms of lesions, and then lacunes, microbleeds, other MRI changes. Um, so yes, so sorry, slide 4. So what kind of brain changes can CADASIL produce? So specifically, white matter injury, small fluid filled in spaces, tiny microbleeds or cerebral microbleeds, um, changes in diffusion, um, or really white matter organization, and then tissue loss. So collectively, all of this together is what we would call disease burden, and they don't all mean the same thing. Just I really want to stress that. Um, so MRI is effectively a window into visualizing and seeing all of this together as disease burden. But what it doesn't do, at least yet, is give us a score. We don't get like one value. Oh, you've got this. This is your score. Many diseases are this way, where you just sort of get a number and that's where you're at. So we do get either totals or summaries or even regional summaries for each of these white matter changes, but taken together, or just even just simplistically totaled or combined, it still really doesn't give you a single score that you can use. So what it does give us, though, and next slide, 6, please, is it does give us a way to visualize and understand patterns of brain injury. It gives us the ability to track those patterns and those changes over time. It gives us the ability to compare and contrast the global and regional burden scores. And it serves as a way for clinical trials to use aspects of these features and measures when designing those trials, when conducting those trials, when assessing the results. What MRI can't do, though, is perfectly predict one participant uh, what their future might look like. It doesn't explain really any of the symptoms that you might see on their own. It doesn't replace your clinical story, and it doesn't define you as as a person. So, and I love this sort of message. So I just really want to state this just as strongly as I can. It's informative, but it's not your destiny. Um, Next slide, please. Um, so how do we turn what we collect as MRI into measurements? Um, and that's my job. I help with this. So I help with the methods to do this. I help with finding other teams ways of doing it and integrating that or extending it or improving it or building on our own sort of tools for doing this. And so we start with identifying what are the MRI visible markers, and then how do we segment or map or just identify that uh, quantitatively exactly. And then so that leads to a map or just a sort of in every spot in the brain, you know, is this tissue white matter, gray matter, CSF, blood, etc., or is it a identifiable marker such as a lesion or lacoon or a microbleed, etc.? And so that's what our sort of our containerized pipelines or our measurement tools, that's really what they're doing, is they're saying it's either um, it's either one of these anomalies or it's normal looking or normal appearing brain tissue. Next slide. Should be number 9. So I want to take you through just the foundation of uh, what the CADASIL imaging markers look like. And, while we'll use the white matter hyperintensity or white matter lesions as the anchor, I want to take you through just beyond that, and then how combining or using sort of all of these markers together will lead to clinical trials and essentially monitoring disease progression and potentially starting to um, predict future outcomes. Next slide should be, I don't know, it says 7 on mine, but it's disease over time, conceptual timeline. Yes, thank you. Okay, so as I stated earlier, the MRI changes can actually appear really before anything else, at least symptom wise, may appear. And most, if not all of them, are going to gradually change over time. And this is labeled as accumulating burden, but what we mean here is that um, where you might see lesion will start to expand um, laterally away from sort of the center of the brain, moving towards like either to the front and back and midsection of the brain. Um, and then also as time progresses, you'll start to see um, symptoms and changes in function. Um, Next slide. So on the right here, you see just kind of a cartoon of really what each of the white matter sort of anomalies or changes in white matter um, sort of look like and where they're sort of located. So, um, and I'll go through each of these in detail, but the white matter lesions themselves can be found really in the subcortical regions and cortical regions. Um, you can see enlarged perivascular spaces detailed here. Oh, I forget. Sorry, you can't see my mouse, so I won't use my mouse. You can see microbleeds as well as lacunes. And so again, we track this sort of globally, as in how much of each is there, and then also regionally, where is it happening the most? And over time, where is it changing the most? Um, so next slide should state introducing. Yep, thank you. All right, so what is a white matter lesion? And why is it called white matter hyperintensity? And so this is just the hyperintensity is just uh, a term for the uh, quantitative signal value that the white matter has on, in this case, either a T2 or flare image. And hyper intense just means that it's brighter than it should be. Um, and so what we call lesions on flare images look brighter where they shouldn't be bright in sort of intensity value. And so because they're brighter than what should be expected, various image analysis tools have either learned or been trained or utilize AI to essentially automatically or semi-automatically classify or create a map of where lesions are in the brain. And so in this kind of cartoon example, um, where you see the bright areas, and trying to see if they're not circled in this one, but I'll show you another slide here in a bit where the actual lesion classification is circled. But essentially all of this bright area is actual lesion. Next slide, please. So the white matter lesions give us a strong anchor for explaining disease burden. Um, they're easy to visualize. They're often extensive, and they can be summarized globally and regionally. Um, and so next slide. Yes, so it's as we've stated, the sorry, the lesions themselves are kind of the anchor, but they're not the whole story. And the reason that I sort of frame it that way is if you look at how much of, say, the clinical symptoms, functional outcomes, uh, cognitive testing, etc. If you look at, if you compare like the lesion burden alone to any or all of those other things, it doesn't statistically explain all of the variance, right? Which means that there are other things that, when combined with white matter lesions, help to tell, you know, more of the story. And so that's really what we're trying to do with this landscape of all of these MRI markers, is we're trying to bring them all together to try to explain the symptoms that we see within CADASIL. So as promised, uh, this is just an example of actual uh, automated classification of the white matter lesions. And so I'm comparing on the top row, I'm comparing a um, participant that has a low amount of white matter lesion or white matter burden compared with a participant that has a high amount. And so the areas that you see um, circled that are bright are all what we would call lesion tissue. And so that's actually what we're measuring. And the amount that we measure is essentially normalized or divided by the total brain volume. So you're looking more at like a ratio of lesion over the entire brain size. And you can do that as one value, or you can do that sort of in every meaningful region of the brain. So that could be the lobes of the brain, the subcortical areas of the brain, the cerebellum or midbrain, pretty much any region that you want to look within. You're able to sort of quantify the amount of lesion within that region, um, and then compare it to other regions, and again, or the total brain. Um, so next slide, please. Should be 12. So why do the regions matter? Well, two people can have the same total burden or the same, amount of total white matter lesion over their brain size, but where the lesions are in the brain may differ. And so that alone could explain different symptoms and a different sort of timeline or disease progression, or even disease expression. So we want to look at both. We want to monitor both the global sort of value as well as the regional values. Next slide. So where we look is very important, and we look in the deep white matter areas, the periventricular areas, and then both the frontal and parietal lobe. And that's just a small number of the regions, but so far those have been the most important regions that we've looked at. Next slide. So this is one of the actual examples of the data that we've collected. And so this is actually it's considered a heat map, but what it's showing you is every participant, every lesion that we've found. And if another participant had a lesion in that same area, it gets brighter and brighter and brighter. And so we go all the way up to, in this case, 461. That's the number of participants in the study that had lesions of any kind. And as you get brighter and brighter, you can see the most common regions or the most common areas. Now, this data or this heat map is loaded on what's called like a standard or average brain. Obviously, everyone's brain is a different shape and size, but we sort of warp each individual brain onto the standard and then bring their individual lesion map along with that. And so that's how we're able to sort of add these all up and quantify this in the way that you're seeing. Um, so, sorry, next slide. Uh, so one, at least in the literature, one of the most prominent findings has been in CADASIL has been the anterior temporal and external capsule areas. And so I just wanted to highlight those. We of course are seeing that same pattern in the data that we've collected so far, but I do want to make it clear that, or at least be cautious here, only having like anterior temporal and external capsule white matter lesions isn't all of the story. And so even though this one is, the presence of a lesion there is, significant, you know, we also want to look at all the other regions. So I just wanted to make that um, very clear. And next slide. So conceptually, um, we've stated that the amount of lesion changes over time. So we can see that in two ways. The first is just that we follow the same person 2, 3, 4 times. Um, and over time, the amount or total burden, if you will, is going to increase. So in this cartoon example, it shows um, for various ages, um, the amount of typical white matter burden seen at that point, at that age. And this actually starts to exponentially increase in the mid 50s onward. And so it's slow to get started, but then it definitely starts to get increase further and further and further. And we took it, next slide, please. So this is yet another example. This is again our data. And so we are seeing this pattern. For those of you really paying close attention, that the shape of that curve doesn't look the same. Well, it's only because I've put this on a log scale, basically. So you can see from 1 to 10, from 0.1 to 1, so this is exactly the shape of the curve you'd see when you put that on a log scale. And what I did different here, though, is I've divided this into several curves. One curve for our gene negative family controls, and then 3 curves for Notch3 mutation carriers. And we absolutely see a different pattern over time, uh, depending on where your mutation is. And so there's a publication, Hack and all, that divides the mutations into 3 risk categories. And so we matched that and just plotted sort of the lesion burden that we're seeing. And so the rate of accumulation of burden is definitely distinct. And while they all, eventually lead to the same total amount of burden, uh, the the amount of time that it takes to get there is accelerated depending on where the mutation, the Notch3 mutation, is located. Next slide. So white matter hyperintensity lesions can relate to several clinical dimensions, um, but we don't necessarily want to ever state that the lesion is what's causing that clinical symptom or clinical finding. They're associated with each other, but we've got a lot of work to do before we say one causes the other. But what we do see strong association wise is that cognition, gait, processing speed, um, all are associated with lesion burden, and each sort of is changing over time. And then I did just show just how risk stratification, and that's just essentially just means the difference between the low, medium, or high risk Notch3 locations. Um, Next slide. So some limitations of the white matter lesions are um, really good to sort of uh, uh, discuss here, and that's that the lesions themselves don't capture tissue loss at all. Um, they don't in any way reflect on the microbleeds. Um, they miss subtle signs of microstructural changes in white matter, and they don't explain all of the clinical symptom variation. So, with that in mind, um, we are assessing, monitoring, and collecting the ability to look at other white matter anomalies. And so, next slide. So, now we'll just walk through a few of those. The first one is perivascular spaces. And what these spaces are fluid filled spaces around the small vessels. And very important point is that everyone has these. And so it's not enough to just sort of count them or say, oh, you have enlarged perivascular spaces. Um, what you want to do instead is you count them, you sort of regionalize or localize them, and you can measure the size, or at least the amount of each one um, in relation to all the others. And we do know that they become more visible or more enlarged uh, when the brain is under stress. And so one thing that we've seen in our own data so far is that, again, it's not the number of them, it's not their total size if you add them all together. It's not even sort of where they are, but it's the variability in their size that seems to be most important. So we are tracking that. Next slide. Um, and this, again, just a borrowed figure, but this just shows examples of just different sort of locations and patterns and levels of the enlarged perivascular spaces. Next slide. Um, so the next white matter finding that we are tracking are lacunes. And lacunes are small cavities left behind by um, a prior small stroke or infarct, and they are visually apparent in that they're sort of like a small black hole. So you could see here on this flare image that where the arrow's drawn, first off, you can see a rather large amount of white matter lesions on both sides. And then this location right here is a lacune. And so, um, next slide. Oh, we'll go back to lacunes. So we'll just talk then about so for lacunes, what we do is um, we count them. We do measure each lacune's volume. We add those all together. We track where they are regionally. And for lacunes, at least so far, what we've seen is that, like white matter hyperintensity, the total volume is probably the most important, and then also the count itself. How many of them are there? Um, Unlike the perivascular spaces, the number of lacunes is actually, uh, I think, clinically relevant, and so we'll uh, be using them in that sort of context. So next slide. So microbleeds, cerebral microbleeds are next on the list here, and the arrow shows one, and you can't see my mouse, but if you work with just the one dot that you can see, you'll see that there's, you know, 6, 7, 8 of those on each side. So each of those is a microbleed. And in this case, the microbleed's size is not as important as just the sheer number and also the location, but the number of them is probably the most important. And we are very careful on the size only in the context that if several bleeds sort of land on top of each other, it looks as if it's, you know, a much larger bleed than other bleeds. And so we actually call that a macro bleed, and we can separately count those. But as I said, they're usually just that, given the way that we're imaging this, they're just so nearby that they look like they're the same bleed, but it's just more than one bleed adjacent. Okay, next slide. So peak skeletonized mean diffusivity. This one is fun. And really what this is that diffusion imaging essentially tracks the ability for water to move freely um, through white or gray matter. And white matter is much of it is fat. And so that means that it's hard for water to move through it because fat and water don't like each other very much, right? And so when the structure of the white matter is changing and potentially has less fat, less organization, that means it starts to become more, it allows more diffusion to occur. And so we can measure that. And so on the top row, um, you see um, a control image where you can see that within the white matter, um, there's very little diffusion occurring. You do see uh, much more diffusion occurring in gray matter, and you would expect that. And then within the ventricles, where CSF is, that's water, and so you can see the maximum sort of diffusion occurring in those regions. Now, in a CADASIL participant, you can see that the diffusion within white matter is definitely changing, and it's more it's allowing more diffusion to occur. And in this method that we're using, we actually map the diffusion itself onto the main sort of tractography. And on those tracks, we actually then are able to calculate the diffusion on the track itself. And that is what this marker shows. And you can see that it's the PSMD itself is the value that we get, this peak skeletonized mean diffusivity, is reduced in the CADASIL in comparison to the control. So we are seeing that same pattern in the data that we've collected so far, and we'll be reporting that hopefully later this year. Next slide. So atrophy is usually characterized as the loss of gray matter, tissue. And so we do see this pattern as well. It's accelerated atrophy. It's regional, and it also contributes really to the overall changes that we want to sort of track cross-sectionally and over time. Next slide. So we've kind of gone through each of these items, and I just want to kind of put it all together now and say that really to tell the entire story through the lens of MRI, we need to think of each of these regionally and globally, and then how they sort of fit together. And that integration is, I would argue, going to be much stronger than any one marker alone. And so that is really going to be the nature of our work within our consortium, really over the next year or so, is that first we're going to report the findings on each of these items, and then we're going to put it all together and then report just how, when combined, uh, you know, how like any sort of combination might better explain the variation that we see in clinical symptoms, functional outcomes, and really quality of life. So next slide. So yeah, this just says the same thing. Um, so next slide. So why do these markers matter for symptoms? So we want to look at each marker and then look at how it relates to cognition, um, clinical events, um, functional outcomes, and even other biomarkers such as the blood-based biomarkers that we've collected and any of the assays that we're able to run from the blood biomarkers. Next slide. So what does this mean to um, participants, patients, families, um, and to start to just frame some of the questions that you may have, you know, does having these white matter changes mean that your symptoms are going to be worse? And can MRI predict your future? And why do some family members, maybe even with the same mutation and same environment, um, differ? Um, these are all questions that we have in our minds and that we are trying to organize our research to help start to answer. But as of now, we don't have answers to these questions. Um, so, next slide. So, for longitudinal research, um, we really want to make sure that we're able to assess the changes in each of these markers and when and where they occur. And we want to make sure that the way that we do this, the way that we generate these measurements is reliable over time. Because without that, it makes them difficult or not possible to be used in clinical trials. Next slide. So good biomarkers um, allow for detection of treatment events, or sorry, treatment effects, right? And so oftentimes what you want to show is when given the treatment, um, the expected sort of change in that biomarker is deflected downward, or just that uh, you essentially have to show that it um, that the endpoint itself, you know, changed less than it normally would in a sort of meaningful way, that is, quantitative, valid, and can be repeated and seen across several individuals. Um, so next slide. So one thing that I probably didn't stress as much, but I should have, is just that across those various white matter biomarkers that we've discussed, when we sort of first see those, and how the rate that they change, and sort of when they sort of stop changing, all of that is variable, but also has a at least somewhat of a pattern, in that we see the white matter lesions sort of first, and then we see changes in, like, the lesions at the anterior temporal pole. Um, we see lacunes and tissue loss, much later in the disease, and we see microbleeds actually can kind of change or appear really anywhere along the way. So I just want to make the case that each of these sort of biomarkers on their own have different timing and rates of change, and all of that has to be factored in and tracked, and also looked at longitudinally within the same sample, which is uh, the design of our CADASIL consortium, is set up to do that. Next slide. Um, Another thing that it's just helpful to have in mind is just that um, there are sort of preclinical, early clinical, um, active, and then advanced progression markers. And so we are assessing sort of each of these. It's if you are a participant in the study, this is why we do exhaustive questioning on migraines with aura, um, exhaustive cognitive testing, and functional testing, etc. We again, at each sort of phase, we want to be, we call this deep phenotyping, but we want to be sort of collecting a little bit of everything all along the way so that we can integrate that with these imaging biomarkers, as well as, again, the plasma and other wet biomarkers that we either currently collect or plan to collect in the future. Next slide. Um, so standardization of generating these markers is incredibly important. Um, we want to have the same marker have the same meaning, regardless of like where it's collected, how it's collected, how it's analyzed, and uh, eventually um, be able to collect it sort of anywhere and generate the same sort of marker result. So part of what we're doing in our consortium is that very thing. We're trying to standardize how we're collecting the MRI biomarkers, how we're analyzing them, and then trying, or well, we will be sharing how we did that with the larger community so that um, other research studies can adopt and follow our procedures. And then hopefully pool, share, and collectively analyze even larger data sets together to get ourselves ready for trials. Uh, so next slide. And this is just, the standard way that you prepare for clinical trials. Uh, you measure the changes, you stratify the risk, um, you select the outcomes, and then you um, ultimately improve the probability of having a successful trial. Next slide. So what can you take away from what I've tried to impress on you in terms of just what are the scan findings and what do they mean on their own and how can they be integrated? So we really want to try to take or move away from isolated individual findings and move towards an integrative approach where we look at all of the biomarkers, omics, deep phenotyping together all at once. To me, this has the best chance at um, establishing and qualifying biomarkers and being sort of ready for treatments. Um, so next and near the final slide. So CADASIL produces measurable patterns in brain injury using MRI. And white matter lesions are central to this, but they're not the whole story. Um, I would make the case that it's the regional patterns and um, the regional patterns together with total burden that really tell more of the story, and integrating all of these biomarkers together is going to better support um, progression tracking. Um, so next slide. So yeah, and just more of this, more to this is just that pairing the global and regional measures together um, is gonna hopefully help us better explain the clinical outcomes and following all of the above over time to detect changes over time um, will occur, and harmonizing how we collect, measure, calculate across sites and across projects, and hopefully or eventually even out, worldwide, or at least collaborative with European groups, Australian groups, um, Southeast Asia groups, et cetera. We want to sort of work with everybody to harmonize and hopefully integrate and be able to sort of look at all of this together. And as said, it's creating this treatment ready data set in partnership with our patients and families that um, will really let us be ready for um, treatments that hopefully come. So I want to thank you all for your attention, and I would love to answer some questions. And Elizabeth, I'm not sure how I can look at the questions.
Back with
So
you. Thanks. I'm going to see if we've got time, and I'm also going to post where to send your questions if we don't have the opportunity to address them. So let me
yeah,
just,
because I'm more than happy to answer any questions now or after today. So please feel free to reach out. I'm trying to think, what typically do we do email address wise? Do we have a dedicated sort of
yes, and I'll
location
post
that?
that here.
Okay, great. Thank you.
In fact, yeah, I'm going to post that now because I don't have pending questions here. And I think I have always promised to kind of finish on time. So with that, I think I'm going to ask that people check where I've posted in the Slido app. And any questions for Jeremy, we would be happy to take. And let's just see, you can see the email address down below, info@cadasil-consortium.org. And we will get to those as promptly as possible. So please send your questions there. And Jeremy, thank you so much. I think it's actually the first time that we've seen, real data from the study presented here in this format. So it's very
Yes, yeah,
compelling and
it was really
appreciated.
exciting that I got to do that. Like um I don't want to say I hesitated. It's just we've got so much in the works. And to all of you, just stay tuned because we're gonna be publishing quite a bit now over the next, 3, 4, 5 months. You're gonna start to see several publications come out from our baseline visits. So
Thank you so much. I hope we can also rope you into another webinar down the line or next year to kind of
yeah,
share those
absolutely.
as
I'd
well.
love to do that, especially when we get to longitudinal analyses. So maybe like a year out or so, or
That would be
9 months
great.
out or
Thank
so.
you for your time. Yeah, well, much appreciation to you. And thank you, everybody who joined us tonight. We look forward to seeing you. I'm not sure if we're having a June webinar because I know we have CADASIL consortium meetings, and then there's also the ULF meeting and a couple of other competing time slots. So we'll keep you posted about that. If we don't see you in June, we will look forward to resuming in July. Have a great evening, everybody, and thank you so much.